Hello everyone. Today's lecture is on efficiency of pasteurization. The main objective of today's lecture is to study the time and temperature combination which is used during the process of pasteurization, efficiency of pasteurization, and phosphatase test. So first, time temperature combination used in pasteurization process. So pasteurization is the process which includes heating of every particle of a milk at a particular temperature for specific period of time to destroy any pathogen which is present in the milk, and at the same time there should be a minimal reduction in the nutritive value of the milk. With pasteurization, some organisms which are present in the milk get killed because of heat sensitive in nature. Some organisms will be inhibited or attenuated, while the spore-bearing organisms which are present in the milk will stimulate to germinate because the spore bearers are the heat resistant. The lethal effect of temperature on the organism will depend upon the resistance of that organism towards the heat. So pasteurization is a temperature time effect. That means this process depends on the combination of the two parameters such as temperature and time, and not on either of them alone. So the criteria which is used for the selection of temperature time combination for the process of pasteurization. So the first criteria includes the most heat resistant pathogen. Mycobacterium tuberculosis was considered previously as a most heat resistant pathogen which causes tuberculosis. But nowadays, instead of Mycobacterium tuberculosis, the another pathogen, that is Coxiella burnetii, which causes Q fever, is considered as a most heat resistant pathogen. And this pathogen, Coxiella burnetii, should get killed when milk is exposed to a particular temperature for specific period of time. So the pasteurization should responsible for killing of this most heat resistant pathogen. Killing of this heat resistant pathogen indicates that there will be killing of all other pathogens, as other pathogens are sensitive to the heat. The phosphatase test should be negative. And the third criteria which is used for the selection of temperature time combination for pasteurization includes the cream line, means the fats which are present in the milk should be minimally reduced. The quantity of that fat should not get decreased. Since the most heat resistant pathogen, that is Coxiella burnetii, is destroyed at a treatment slightly higher than that required for phosphatase inactivation, and that temperature should be below that which is required for the reduction in the cream line. It is observed that lower the temperature of exposure, the longer the time needed, and vice versa. Efficiency of pasteurization. The efficiency of milk pasteurization will depend upon how much there will be reduction in the number of pathogenic organisms present in the milk sample. And this reduction in number of pathogenic organisms during the pasteurization will depend upon the temperature of pasteurization. It will also depend upon the time of exposure of the milk at that particular temperature during pasteurization, and it will also depend upon the kind of bacteria, means it will depend upon the number of the bacteria as well as it will depend upon the type of the bacteria. So the proportion of the total microbial load that are spore formers or thermogenic organisms. Thermogenic organisms means the organisms which can survive at pasteurization temperature. So the efficiency of pasteurization will depend on the temperature which is used during the pasteurization, the exposure time, as well as it will depend upon the kind of the bacteria, means the number of the bacteria and the type of the bacteria present in the milk. Shelf life of the pasteurized milk will depend upon at what temperature that milk will be stored after the pasteurization. So the shelf life will depend upon the temperature of storage, and it will also depend upon the number and types of the microorganisms which remain viable after the process of pasteurization. Pasteurization does not sterilize milk. The temperature which is used for the sterilization is 100 degrees Celsius or more than 100 degrees Celsius. But in pasteurization, the temperature used is in between 60 degrees Celsius to 80 degrees Celsius, which is less than the temperature which is used for the process of sterilization. So, pasteurization does not sterilize milk, it only reduces the number of the microorganisms present in the milk. The surviving organisms must be kept from multiplying by constant refrigeration. The surviving organisms are readily heat-resistant thermogenics and require higher growth temperature. At refrigeration temperature, the surviving organisms are unable to grow, so they are unable to spoil that milk. Alkaline phosphatase test for pasteurized milk. So pasteurization is an essential process in the production of milk which is safe and free from the pathogens. There are the three methods of pasteurization. The first method is low temperature holding pasteurization or low temperature long time pasteurization, in which the milk is exposed to a temperature of 63 degrees Celsius for 30 minutes, and after heating there should be immediate cooling of that milk at refrigeration temperature. The second method of the pasteurization, called as HTST method, that is high temperature short time method, in which The exposure temperature is 72 degrees Celsius for 15 seconds. While the third method is UHT pasteurization, that is ultra high temperature pasteurization, in which the milk is exposed to high temperature, that is in between 115 to 130 degrees Celsius for 2 to 6 seconds. So alkaline phosphatase is a thermolabile enzyme which is naturally present in the raw milk. It is a thermolabile enzyme, so that enzyme is destroyed at a temperature just near to the pasteurization temperature. Alkaline phosphatase test is used to indicate whether milk has been adequately pasteurized or whether it has been contaminated with raw milk after pasteurization. So during the pasteurization, milk is heated at a temperature in between 60 degrees Celsius to 80 degrees Celsius. At this temperature, the phosphatase enzyme, which is naturally present in the raw milk, is get inactivated. So after pasteurization, if that milk contains the phosphatase enzyme, then it will indicate that the milk is not properly pasteurized. Means milk is exposed not to that particular temperature in between 60 degrees Celsius to 80 degrees Celsius. So this test, the detection of the phosphatase enzyme, is based on the principle that the alkaline phosphatase enzyme, which is present in a raw milk, liberates the phenol from disodium para-nitrophenyl phosphate, which is a substrate for that phosphatase enzyme, and that enzyme converts this para-nitrophenyl phosphate into a yellow color complex at alkaline pH. This yellow color complex is phenol. The intensity of yellow color produced is proportional to the activity of the enzyme, and this intensity can be measured by direct comparison with standard color disc in a Lovibond comparator. Detection of phosphatase is not applicable to sour milk and milk which is preserved with chemical preservatives. So the reagents which are used for the detection of the phosphatase enzyme include first sodium carbonate bicarbonate buffer. Dissolve 3.5 gram of anhydrous sodium carbonate and 1.5 gram of sodium bicarbonate in 1 liter of distilled water. Second reagent is buffer substrate solution. Dissolve 1.5 gram of disodium para-nitrophenyl phosphate in 1 liter of sodium carbonate bicarbonate buffer. This solution is stable if stored in a refrigerator at 4 degrees Celsius or less for 1 month, but a color control test should be carried out on such stored solutions.
Procedure: take 3 test tubes and label it as a positive control test and negative control. In all these 3 tubes, add 5 ml of buffer substrate solution. Then add 1 ml of raw milk in a tube which is labeled as a positive control. This raw milk contains the phosphatase enzyme. Then add 1 ml of test milk in a tube which is labeled as a test and 1 ml of boiled milk in a tube which is labeled as a negative control. So this boiled milk will not contain the phosphatase enzyme because it is thermolabile enzyme. After addition, then incubate all 3 tubes in a water bath at 37 degrees Celsius for 2 hours. After 2 hours of incubation, read the color developed using the comparator. So, after incubation, if yellow color will appear in a tube which is labeled as a positive control, since it contains the raw milk and that raw milk contains the phosphatase enzyme which acts on the substrate and convert it into the product that is para-nitrophenol which is yellow in color. No yellow color will be developed in a tube which is labeled as a negative control as it contains 1 ml of boiled milk. Boiled milk does not contain the phosphatase enzyme, so there will be no formation of yellow color para-nitrophenol. So no yellow color will be developed in negative control. While the color which is developed in a tube which is labeled as a test, it depends upon whether that milk is properly pasteurized or not. If yellow color will be developed, then it will indicate that that milk contains the phosphatase enzyme, means that milk is not properly pasteurized. No yellow color development indicates that that milk does not contain the phosphatase enzyme, means that milk is properly pasteurized at a particular temperature for a specific period of time. Milk that gives reading of 10 microgram or less of para-nitrophenol per ml is considered to be properly pasteurized.
So alkaline phosphatase enzyme acts on the substrate para-nitrophenyl phosphate. This substrate is colorless in alkaline as well as in acidic medium. Enzyme converts this substrate into the product that is para-nitrophenol plus H3PO4. This para-nitrophenol, it is colorless in acid medium while yellow in alkaline medium. Detection of the phosphatase enzyme in the test pill can be with the help of a reagent called as 2,6-dichloroquinone fluoramide. So this is a second test or the alternative test which can be used for the detection of the presence of the phosphatase enzyme in the milk. So phosphatase enzyme liberates the phenol from phenyl phosphoric ester. So phenyl phosphoric ester is get converted to phenol and this conversion is carried out by the phosphatase enzyme. This phenol will then convert it to the indophenol blue, which is blue in color, after addition of the CQC reagent, that is after addition of 2,6-dichloroquinone chloramide reagent. So this test can be carried out again by taking the 3 tubes, label positive control, test and negative control. Add 5 ml of buffer substrate solution in all these 3 tubes. Add 1 ml of raw milk in a tube which is labeled as a positive control. Raw milk contains the phosphatase enzyme. Add 1 ml of test milk in a tube labeled as a test and 1 ml of boiled milk in a tube labeled as a negative control. Boiled milk will not contain phosphatase enzyme because that phosphatase enzyme is a thermolabile enzyme. After addition, then incubate all 3 tubes in a water bath at 37 degrees Celsius for 10 minutes, and after incubation, then add 6 drops of CQC reagent in each test tube and observe the color change. Now the positive control contains the raw milk, means it contains the phosphatase enzyme. When phosphatase enzyme will present, it will act on the substrate, convert it into the phenol, and after addition of the CQC reagent, that phenol will get converted to the indophenol blue. So blue coloration will appear after incubation. In negative control, the boiled milk is used, in which the phosphatase enzyme is absent. So, in negative control, the phenol will not be produced, and due to which no blue coloration will appear after the incubation. While the result of the test will depends upon whether that milk contain phosphatase enzyme or not. If that milk contain the phosphatase enzyme, then there will be the blue coloration, which indicates that that milk is not properly pasteurized, means that milk is not exposed to a particular temperature for a specific period of time, which is used for the pasteurization. Absence of blue coloration after incubation indicates that that milk does not contain phosphatase enzyme, means that milk is properly pasteurized at particular temperature for specific period of time. Significance of phosphatase test. The phosphatase test is used for the determination of efficiency of pasteurization. The contamination of that pasteurized milk by raw milk could result in reintroduction of pathogen. Therefore, it is important that post-pasteurization contamination tests are undertaken regularly. So the references that you have to use for this phosphatase test. Possible questions. State true or false. Phosphatase is inactivated at pasteurization temperature. Phosphatase test is carried out before pasteurization of milk. Fill in the blank. The substrate used in phosphatase test is dash. Explain phosphatase test. Describe phosphatase test and give its significance. Or justify phosphatase test used for determining the efficiency of pasteurization. Thank you.